To the class 1 binding prediction tool, and now let's go through the class 2 binding prediction tool. So before I start, I just want to stress some things again which were already mentioned by others. So the first thing is, if you want to find epitopes in your protein of interest, you should first check if there are any experimentally verified or confirmed epitopes in the database. Then you go to the prediction tools. The second is, the prediction tools don't give you confirmed epitopes. It only gives you potential predicted candidates, epitope candidates. So they are not verified. So some people misunderstand and they take these predicted epitope candidates as real epitopes. So that's not the case. We are only providing predicted epitope candidates which needs to be experimentally verified to be sure that they are epitopes. And thirdly, so we have got your questions. So we hope that most of your questions will be addressed by these presentations. And if they are not completely answered, we have individualized sessions in the afternoon. We can ask, we can discuss them. And if you have questions in future, please email us. And we charge only very less amount, like $1,000 per request. Just kidding. So please email us. We are happy to help you in any case. And also please let us know if you want to have additional features or tools that you would like to have here. So we have some things in pipeline, some tools and analysis tools in the pipeline, but we will try to incorporate your suggestions. So let's start. So I have a brief outline here what we are going to cover today. So first I will start with a brief comparison of class 1 and class 2 tools. And then we will go to the overview of the class 2 tool with some example and descriptions. And along the way I will also be giving the guidelines and different recommendations, threshold values. And we have different versions of the tool. So editor showed you only the web or online version. So we have also the standalone and also IEDB API or RESTful interface. And on the way, I will also show you how we can use this tool to predict promiscuous binders. And recently we published a paper on identifying the top immunodominant epitopes based on that we identified a method designed based on the immune response values rather than binding affinity values. So I will explain that. And I will also take you through other resources available, help, download, etc. And we will have a brief, small exercise. And then at the end, we have now developed a new interface for the class 1 and class 2 tools. So I will give you a preview of that tool at the end. So first, let's start with the comparison of the class 1 and class 2 tools. So the basic structure and principles are same as the class 1 binding prediction tool. So I'm not going to explain in detail, which editor already did, but I will stress where there are some differences and some important things that you should be keeping in mind. So the basic difference between class 1 and class 2 tools actually starts from the structure of the class 1 and class 2 molecules. So if you look at here, this is the structure of class 1 molecule and this is the class 2 molecule. So for class 1, there's only one polymorphic chain, that's alpha, and the beta is constant, beta 2 microglobulin. On the other hand, for the class 2 molecule, you have two polymorphic chains, both alpha and beta, and both these chains impact binding. In case of class 1, there's only alpha chain that impacts binding. And in case of class 1, the binding groove is closed. And on the other hand, the class 2, the binding groove is open. So what difference that makes is that class 1 molecule can bind comparatively smaller peptides, whereas in case of class 2 molecule can bind longer peptides since the groove is open. And so based on the chains, this is a slide for showing the difference in the nomenclature between the class 1 and class 2 molecules. So for class 1, we generally mention only alpha chain. So for example, we say HLA-A or HLA-B or 702. But in case of class 2, for DP and DQ locus, we mention both alpha chain and beta chain. So for example, DPA O103 and DPB O201. But it's mostly that the B chain that we take into consideration when we do the binding predictions. But in case of DR locus, there's only beta chain that's variable polymorphic, and we mention only DRB in case of DR locus. So compared to class 1 binding prediction tool, class 2 binding prediction tool, developing and designing a class 2 binding prediction algorithm, there are some more challenges. So for example, in case of class 2 molecule, there's a 9 amino acid binding core that actually binds with the MHC molecule. So when we design the algorithm, prediction algorithm, first we need to identify this binding core, this 9 amino acid binding core. So this is, for example, if this is a 15-mer peptide, there's a 9 amino acid binding core. And the challenge is to correctly identify this binding core. So this needs proper alignment of the binding core with the MHC molecule binding groove. And another one is the influence of the peptide flanking residues. So you have this binding core, 9 amino acid binding core, and flanking residues. So these flanking residues that flank the binding core also influence the binding in case of class 2. So you need to take this also into consideration, the length and composition of the peptide flanking residues also into consideration when you design a class 2 binding prediction algorithm. And some other challenges in case of class 2 is that the availability of uniform, experimentally verified, measured binding data. So compared to class 1, we have much less data for class 2 alleles. And because of that, we have fewer alleles that are available for class 2 prediction tool algorithms. And because of all these reasons, the prediction accuracy for class 2 is little less compared to class 1. It's not bad, but compared to class 1, class 2 prediction performance is a little bit less. So for example, you can see in MHC PAN, it's 0.82 AUC. In NetMHC 2 PAN is little less, 0.78, and similarly for SMM and SMM align. And another difference between class 2 tool and class 1 tool is that in case of class 1, you can do the prediction for different peptide lengths, for example, from 8 to 14. But in case of class 2, the peptide length is fixed at 15. That doesn't mean that MHC class 2 molecule can bind only 50 mers. No. That's the peptide length that's fixed for the tool, just for the tool. It's not a feature of the molecule, but the tool. I will explain why that's fixed like that later. And since the prediction efficacy is a little bit less, and for various reasons, we have set a higher threshold for class 2 prediction tool compared to class 1. So I will explain the thresholds in the coming slides. So the tool can be accessed from the homepage, IEDB homepage, like editor showed for the class 1, or you can directly access from this link, tools.iedb.org, MHC2. And this is the web interface for the class 2. It's exactly same as class 1. So at the top, you get all the tabs for all the other resources, help, example, reference, download, and contact. And this is where you paste the sequence, copy paste the sequence, and you can choose the method here, allele, species, and locus. And for class 2, we have got only human and mouse. For class 1, you have like 7 or 8 species available, but for class 2, we have got only human and mouse available. And here you can choose the allele. And here we have given a link for going to the NCBI browser for selecting the sequence. So you can just go to the NCBI and find the sequences. It doesn't automatically capture the sequence and paste it here. No, it doesn't do that, but this is just for going to NCBI and finding the sequences of interest. So once you have your sequence, you choose the prediction method. So similar to class 1, we have different methods available here. So these are the methods that we provide. NetMHC 2 PAN and NN align, these are based on artificial neural network method. And SMM align, which is a stabilization matrix method. Combinatory library, Stoniolo. And then further, we have got consensus. So this is same as class 1. Consensus method is a combination of 3 methods, SMM align, NN align, and combinatory library or Stoniolo. And we have this option of IEDB recommended, exactly same as class 1. So if you choose this is what we recommend. If you choose this, if any of these algorithms are available for that particular allele, it will use the consensus method. And if it's not, then it will choose NetMHC 2 PAN by default. So the first question that may come to your mind is like, okay, we have all these methods, what method should I use? So what we recommend obviously, explicitly, it's the IEDB recommended. Just use the IEDB recommended. And as I mentioned now, it employs consensus, which in turn uses combination of NN align, SMM align, and combinatorial library or Stunilo. And if any of these, if none of these algorithms are not available, then it will use NetMSC 2PAN. And the advantage of using this is that these are the best available methods so far, and this gives a consensus percentile rank. I think Edita just mentioned that, but I will explain that in further coming slides. So it gives a consensus percentile rank, which helps you compare different peptides and across different methods. And in case you want the predicted binding affinity and percentile score for the component methods, individual methods, it also provides that. It gives the binding affinity and percentile rank for each method separately. So now you have your sequence and method. You chose the method. Next one is selection of alleles. So first you choose the species and locus, and then you have several options for choosing the alleles. So first one is the most frequent alleles. So by default, we provide in the drop-down menu the most frequent combinations of, in case of DP and DQ, the most commonly occurring combinations of alpha and beta chains. So that's by default. You can just click on that, just similar as class 1, you can click on that and select the alleles. And in case for DP and DQ alleles, if you want to select the chain separately, so by default we provide the most commonly occurring combination. So if you want to select the alpha and beta chain separately, you can check this box here, which gives you option of selecting alpha and beta chains separately for DP and DQ alleles. And the next one is the reference set. So similar to class 1, most frequent allele reference set, we have a set of 26 alleles in case of class 2. So by checking this box here, you can automatically select those most frequent alleles. So we can use this in different applications. I will explain that later. So more details on this are available at the help page, section 3. So there's a link towards the allele frequencies. You can refer that. And the last option for uploading alleles. So if you have repeating analysis using the same set of alleles, you don't have to keep clicking. So you can just upload the alleles as a text file. You just enter all the alleles that you want, and you can upload that. But please remember a few things. First thing is, you can do the prediction for only the available alleles here. You cannot just upload any random allele. You can do the prediction only if we have the algorithm for that particular allele. So if you are uploading the file, just remember to check if we have got this algorithm available. And secondly, at this point, you cannot upload the allele sequence. You need to enter the allele name. We cannot accept the allele sequence as of now. We may in future include that feature, but as of now, we cannot accept the allele sequence, protein sequence. And thirdly, you have to follow the specific format for mentioning the allele names. So more details are given at the help page. So just follow the specific format, and please make sure that these alleles are available. So now you have your sequence, you have the method, prediction method, and you have chosen the alleles. Now what happens? So once you submit, let's see how the tool works. So first of all, the sequence breaks down the tool breaks down the sequence into all possible 15 mers. So the peptides will be overlapping by 14 amino acids. And then it predicts the binding affinity for each individual peptide. And then further, it takes this predicted binding affinity of each peptide and compares with a large set of randomly selected peptides. It's same as class 1. And then comparing to this set, it assigns a percentile rank for this particular peptide in question. And then consensus, as I mentioned earlier, consensus employs 3 different methods. So the consensus percentile rank means that's the median value of the 3 percentile ranks from the 3 different methods. So its consensus percentile rank is generated by comparing the selected peptides' predicted binding affinity against that of a large set of peptides. And why we use this? It provides a uniform scale for comparing between different peptides as well as different prediction methods. And a lower percentile rank value. Lower value means better affinity or better binding affinity. In case of consensus, I just mentioned it's the median of the 3 percentile ranks that we get from the 3 different methods. So now let's see an example, and I'll explain how we proceed. So here's the question. So predict the peptides in the NS3 region of hepatitis C virus polyprotein that binds to MHC Class II allele DPB 0401. So how we proceed? So here I got the sequence for hepatitis C virus NS3 polyprotein region. I choose the method IEDB recommended and choose the allele here and submit. So here's the results page. So at the top you have got the input sequence and then the link for downloading the result. I hope it's working now. The bug has been fixed. And then there's a link for citations, which if you happen to use these results in your publication, please cite these references. And here's the results. So this one also looks exactly same as the class 1 results. So you have the allele here, the sequence number, and the start and end position for the peptide, and the 15-mer peptide sequence, and the method that was used. So for this particular allele, we have got algorithm for a combinatorial library, SMM, and NN. So we used consensus. If these were not available, it should have automatically used NetMSC 2PAN. And this is the consensus percentile rank, the last column. And I also want to mention one thing here. So when you submit the sequences, you can either submit a single sequence or a set of sequences in FASTA format, or you can submit like peptides, 15-mer peptides, or other peptides also, set of peptides. So anything is fine. So when you click here, check to expand the result, you get the complete results for all the 3 different component methods. So for example, here you can see the percentile rank for combinatorial library, and this is the binding core for combinatorial library, and this is for the same binding core for SMM align, and predicted IC50 for SMM align, and the percentile rank for SMM align. So for each of the component method, you get individual values. And this is how we estimate the consensus percentile rank. So for this method, this is the percentile rank, 19.59. And for SMM align, we got 0.58. And for NN align, we got 2.57. And the median for these 3 values is 2.57. So that's the consensus percentile rank when consensus method is chosen. So this is the citations that you can cite, use if you are publishing. So, and this is the downloaded result. So by clicking this download result link, you can download it as a CSV file. And when there's too much data, so you must have seen that in the exercise, class 1 exercise, when there are more results, it just displays the text format, in text format. You may not be getting an HTML format, you will just get a text format. And this is CSV that's downloaded. You can use any spreadsheet for further analysis, for filtering and sorting. So now you got the results. So the next question is, okay, I have got all these peptides and binding affinities and personal. So how do I choose the top peptides? So similar to class 1, we recommend here IEDB percentile rank. So just follow the IEDB percentile rank. And the cutoff that we recommend for individual alleles is 10. So percentile rank of 10. So all the peptides that has got a consensus percentile rank or percentile rank less than 10 can be considered as potential binders. And this roughly corresponds to an MHC binding affinity of 1000 nanomolar. So select all peptides with IEDB percentile rank less than or equal to 10. And for our question, the peptides from binders from NS3 region of hepatitis C virus, we have got 23 peptides that binds to this allele with a percentile rank less than or equal to 10. So that's the answer.
A question, sorry. Just looking at taking that median value, for the MHC Class II, there seems to be a much wider range in values. And I wondered if you could comment on that.
Yeah, so we did that, did a study like, so this paper, so we compared different methods and then this consensus method, and we found that if we follow this consensus method, that tends to pick the better peptides rather than picking each individual methods. So that's why we propose to go with the consensus method. Yeah, so we recommend a threshold of 10 here, but I want to expand on that because the recommended threshold is not a fixed value. For example, you get for a peptide, you have 9.99 percentile rank, and for another, there's 10.01, and you cannot say, oh, the one that got 9.99 is a strong binder, and 10.01, it's not a binder. No, it's an arbitrary value. So if you don't have a specific number of peptides in mind, or you don't know, you don't have any idea, that's the value that we recommend. But it can vary depending on your purpose or your study. Suppose sometimes people want, okay, I need more peptides to do my study, then of course you can ignore the threshold and you can select as many peptides as you need. But the scale, you can use the percentile rank scale as a strategy for selecting the best peptides. So you select the peptides with the lowest percentile rank, lowest value, and you get the better predicted peptides. So now you have got the results and you know how to choose the peptides. But there can be an issue here. When you submit the sequence as in an entirety, like when you submit a whole sequence, it's generating the all possible 15 mers. So for example, this is the results that we got right now. So the tool breaks the sequence into all possible 15 mers, and the peptides can be like overlapping by 14 amino acids. So that's kind of redundant. For example, if you look at these peptides, the top peptides, they start at 525, 26, 27, 28, 29. So all these peptides come from the same region. So it's kind of redundant. You don't need to synthesize all these peptides. So what we propose and this is because they all share the same binding core.
Yes. So that might be an advantage for immunogenicity, but it's a problem for predicting what tetramers are going to work in a particular case, because if you don't know the precise frame, you have ambiguity in the tetramers and so forth. So the question is whether you have a way of evaluating which one is better or which one might be better as far as the real immunodominant peptide.
So in general, these core predictions are not necessarily identical with the structural defined core, because there's some ability of those to wiggle around. And we have now actually worked with people in Denmark to come up with a better predictor that keeps the core more stable. So all of these, and this is going to be coming out. Okay, so so the method is always going to be on the website soon. That was actually specifically from John Altman who asked for this and to get these things out. So yeah.
So one way that we use, one approach to exclude these redundant peptides is to do the pre-processing. So what we do is, like, instead of submitting the sequence in entirety, in full sequence, we pre-process that. We generate 15 more peptides overlapping by 10 amino acids, and then submit this set of peptides, 15 more peptides. So for example, if you have a sequence like this, we generate 15 mers like this, and we submit the 15 mers for prediction. So you may be asking why, again, this question, why 15 mers, why overlapping by 10 amino acid residues? So 15 is found to be mostly the preferred length for MHC Class II molecule. It's not the only length, but kind of mostly preferred length. And when you choose 15 mer with 10 amino acid overlapping, you can get the minimum number of peptides with all possible 9 mer amino peptide binding core, 9 mer binding core with at least one flanking residue on both sides. So that's why we are using this length of 15 mer and overlapping by 10. So you can use any Python or Perl script, or even Excel or any spreadsheet for generating this 15 more peptides from overlapping by 10 from your sequence of interest. And another approach, of course, once you did the prediction for your entire sequence, you can do post-processing. That means you can exclude the largely overlapping peptides. So if you have this result here, you do select the top peptide and then exclude the remaining peptides which is overlapping by more than 10 amino acids. So this can be a little bit complicated. So that's why we use mostly the pre-processing method. But by pre-processing, you will always identify the region of interest, and you'll have that binding core. But if you use this method, you are sure to capture the best peptide from that region. So you can either use the pre-processing or post-processing to avoid the redundant peptides. So that's about the prediction for individual alleles using the web interface. So you now know how you can submit your sequence, what method to choose, what are the thresholds, different thresholds for choosing the best peptides from your results. And now let me take you through prediction of promiscuous binders. So you can use the same tool and find promiscuous binders from here. So what are promiscuous binders? These are the peptides that can bind with more than one allele. So most of the peptides, they can bind with more than one allele, and what's the significance? It is found to be associated with stronger antigenicity, and since it can bind with much more alleles, it can have more wider population coverage. So this can be used for finding like the more immunogenic regions, and you can reduce the immunogenicity by mutating this region and reduce the immunogenicity in case of therapeutic proteins. And this can be predicted using the binding affinity prediction. So here one difference from the previous one that we saw. Previously we were doing the prediction for one allele at a time. So there the recommended threshold was 10, but here since we are using multiple alleles at a time, we recommend using a higher threshold of 20 because we don't want to exclude potential binders when we use multiple alleles. So this is only one thing that you have to keep in mind. When we use this for doing the promiscuous binding prediction, you have to use a higher threshold. So how you do that is exactly the same. The only difference is you choose all your desired alleles here, multiple alleles instead of one allele. And then you get the results same as before, but only the thing is all these results for all the alleles are combined here, intermingled. And you have to identify the peptides that binds with more alleles. So now there can be a question, so similar to what you had before, what set of alleles can I use? So if you have a specific population in mind, and you know the specific allele types of that population, you can use that set. And if not, this is the set of 26 most frequent alleles in the global population, similar to what we saw in the case of class 1. So if you can use this one, it's a general most frequent set of most frequent alleles that's seen in the global population. So there's a link provided in the help tab, section 3. More details are given there, and you can download this file also. So for our sequence, so how do we do this? So we find the binders. So binders, for identifying the binders, we have to use this cutoff of 20 percentile rank of 20, and then we have to find peptides that binds with at least 50% of alleles. So that's a recommended threshold here. So out of the 26 alleles, so we have a set of 26 alleles here. So I did prediction for the 26 allele for our sequence, and then there are these peptides bind with different number of alleles here. And if I check the percentage of alleles that each peptide binds, there's only one peptide that binds with more than 50% of alleles. So from this sequence, I get one promiscuous, really true, predicted promiscuous binder. So that's how you can do the prediction of promiscuous binders using this class 2 binding prediction tool. And now there's another.
Did you you had to do that manually?
Yes.
Yeah.
As for this tool, you have to do that manually. Yeah. And now let me take you through another method that we identified recently. We published this paper this year in Journal of Immunological Methods. So here in this study, we used the immune response data from different data sets. Instead of binding affinity, we had 6 peptide data sets with measured immune responses in terms of SFC. And these are the different data sets that we used. There was one from house dust mite, one from Timothy grass, and 2 data sets from TB. And we used a data set from cockroach and pertussis for validation. And altogether, there were around 2,400 peptides and the response data from around 140 donors. So what we were trying to do was to find the method or the approach that will give the minimum number of peptides that can give maximum amount of immune response. So in a way, kind of identifying the predicted peptides, most immunodominant, most potent peptides from the set of peptides from your sequence. So our aim was to capture maximum immune response with minimum number of peptides. So we tried different approaches using the available methods. So we were not trying to design a new algorithm, but we already have different methods, different algorithms and these things already. So we were trying to tweak these already available methods and which approach can give the maximum response using minimum number of peptides. So one approach out of several that we tried, we found that when we used the median, so we used the 26 most frequent alleles and we did the prediction and estimated the median IEDB consensus percentile for these 26 alleles, we found that this was better than using the promiscuity approach, like the promiscuous binder approach. We found that if you go with the promiscuous binder approach, you required approximately 31% of peptides to capture 50% of immune response. But if you go with the median, you needed only 26%. So that was like kind of 5% peptides gain to capture 50% response. And then further we tested, okay, the DP and DQ alleles are not that good, in terms of binding prediction. So we tried excluding those alleles and different combinations of alleles, and we found that if we exclude the DP locus, it improved again the efficacy. So for example, approximately 26% of peptides could capture 50% of the immune response. And further filtering down, screening, we found that if we choose a set of 7 alleles, that's like 3 DRB1 alleles and then 4 DRB3, 4, 5 alleles, a set of 7 alleles and estimate the median, we could get 50% of the immune response using 21% of the peptides. So this is not super performance, but it's better. So approximately 21% of the peptides can capture 50% of the immune response. So this can, and this value, this amount actually roughly corresponded to 20 median consensus percentile rank for the 7 alleles. So this can be kind of called as a universal prediction threshold. So, and we call this as a 7 allele method. So this is published in Journal of Immunological Methods in this year. So how we do that? So first you generate the 15 more peptides overlapping by 10 amino acids, and then do binding prediction for this set of 7 alleles, and then estimate the median percentile rank. So for each allele you get a consensus percentile rank, and you estimate the median, and select all those peptides with median percentile rank less than or equal to 20. That's it. So it's not straightforward, but and you'll have to do that manually as of now. And this set of peptides, all these peptides with median consensus percentile rank less than or equal to 20 can capture approximately an immune response of 50%. So that's it. And do you have any questions so far? Okay. So the other resources is just similar as same as class 1. You have got the tutorial here. Everything is explained in detail here, and you have got example here and references. And the next tab is download, where you can download. There are many things that you can download. First of all, there are like MHC2 binding data sets and prediction scores for MHC2 binding data sets, combinatorial peptide scanning library scoring matrices. So most of these data are of interest for tool developers or benchmarking, people who do benchmarking studies and things like that. And there's also another, so until now I was explaining the online version, web version, and now we have another version, the standalone version. So this is you can download, I think editor mentioned this already, you can download this tool as a standalone version on your computer. So you can download the tool from the same tab, download tab here. And the advantages, so this is available for Linux only, class 1 and class 2 and other processing tools also. Some of the tools currently we are implementing these, like more tools are making available as standalone tools. So right now we have class 1 and class 2 and some of the other processing tools. And this, actually the standalone version runs only on Linux as of now. We don't have a Windows version. So you need to have a Linux. So even if you have a Windows machine, you can have a virtual machine and install Linux inside that. That's what I do. So either VMware or VirtualBox, you can get free copy, and by using that you can create a virtual machine, and inside that you can install Linux. So you need Linux to run this standalone tool. And what are the advantages? So you can implement your the tool on your own machine, and you don't have to depend on internet connection. And the major advantage is in case of large amount of data. So the online version, the web version, will not be able to handle large amount of data. It's designed for only small amount of data. And just now you must have seen, like, when a lot of people are working on the online version at the same time, it may slow down, sometimes it may crash. So standalone tool, you can use it at your own convenience, and you don't need your internet connection. And this is free for non-profit and academia, and there's a license for industry. And also in case of when you have to do repetitive analysis, it's always easy to use the standalone tool. And one thing that you have to keep in mind is that the web version, online version, is not secure. It's just unsecure version, so we don't have any security features implemented. So if you have some proprietary sequence or anything, like you may have to. This happens from industry, so they have this question. If you have some proprietary sequence or something like that, it's always but advisable to use this standalone tool on your own machine, within your environment. So this is a sample command, or how it will look like when you run this standalone tool. So this is a terminal, Linux terminal, and here you can mention the method. So if it's class 1, you just mention MSC 1, and then in case of class 2, and then you mention the method that you want to use, and then the allele that you want to use, and here's the input file for your sequence. And if you want to take the output, the results to an output file, you can specify that here. And all the detailed instructions are available in the README file, which you will be downloading along with the tool. So it's free for academia and non-profit, so you can try it out. And then, so now you saw the online version and the standalone version. We have one more version, API or RESTful interface. So there's this link here. It's not at the class 2 tool page, it's at the main analysis resource page. So there's a tab here called Tools API. So what this does is, actually, it takes your, so both class 1 and class 2 tools can be accessed by this system. So what this does is, it takes your input parameters and sends it to the server at our institute. So it needs internet, but you don't have to use the online interface, web interface. This is useful if you have a pipeline, or you are making a script and you want to do repetitive analysis for some specific alleles, or then it's always useful to it's helpful to use this one. So you just insert your command into your script and you can just run it. So the advantages are, you don't have to install the tools on your machine, where in case of standalone you have to install that, and you don't have to use the web interface. And the requests are sent to the tool server at our institute, and it's freely available to all users, everyone, industry and academia as well. And this is automatically updated. So if you are using the standalone version, downloaded version, then you have to check for updates, regular updates. But this one, it's automatically updated, so you don't have to worry about updating it or reinstalling. And this is a sample command for using the API. So here you specify the method recommended and specify the sequence here, alleles, so you can insert multiple alleles at a time. And so this is for class 1, not class 2. So in case of class 1, you have to specify the length. So here I am using length 8 and 9. In case of class 2, you don't have to specify the length, it's fixed at 15. And this is the URL where the command is being sent, and this is the result. So you get this exactly same result as you use the web interface. And you can take this to output file by specifying the output file here. So that's it. So until now we saw the different versions, the web interface, the standalone version, the API or RESTful interface, and we went through different recommendations, thresholds, and you know how you can choose the top peptides. So right now we'll go through a brief exercise. Here's a question. I'll give you 5 or 10 minutes and you can do it. After 5 minutes we'll go through this together. Okay. Were you able to do that? So the question was, predict the alleles from the given set of 6 class 2 alleles to which this peptide may bind. So the approach is, so you take the sequence and do the prediction and identify alleles with consensus Personal rank less than or equal to 10. So you have your sequence here, and choose the method IEDB recommended and select all the possible, all the 6 alleles here, submit, and you should have got this result. Did everybody get that? Okay, good. And so we have 2 peptides with Personal rank less than 10, and so they are this peptide can bind with these 2 alleles. Anybody any question? Okay, so the next one is TepiTool. So it's a new interface for the prediction of class 1 and class 2 epitope candidates. So we have now, right now we have class 1 and class 2 tools separately. So what we designed a combined interface for both the class 1 and class 2. So the motivation was that we wanted to make it more user friendly, and we wanted to provide all the recommended threshold values. And at the end results, instead of providing like all the prediction values for like as a single file, we thought like, okay, we'll provide the top peptides based on your input parameters. And so this is designed as a step-by-step wizard kind of tool. It involves 6 steps, and it provides the recommendations as default values, and the input parameters which are given us by default can be adjusted as your need. And the new methods that I explained just a few minutes back, like the 7 allele method, all those have been incorporated into this one. So we have submitted this paper, so right now it's under review. So once it's published, the tool will be available at this link. So the class 2 tool is tools.iedb.org MHC2. So instead of MHC2, TepiTool. So the tool will be available at this link. So right now I'll just give you a preview or a quick tour for this tool. So here you can see the interface. So this is the basic design is same. At the top you can find all the different tabs for help, reference, download, contact, and you have got 6 steps here. So at the first step you provide the sequence, your sequence of interest or peptides of interest, and you have 2 options. Either you can copy paste the sequence or you can upload the sequence file here. And once you have your sequence ready, you go to the next step. And the second step, you choose the species and allele class. So it's same as before. So here you have the option to choose from 7 different species. And for human and mouse, you have the option of choosing between class 1 and class 2. For all the other species, you have got only class 1. And once you do the selections, the current selections are shown at the right side panel. And once you have your selection done, go to the next step. And here you have the option of specifying the alleles. So here we have chosen human class 1. And now at the first option provides a list of alleles that are mostly frequent or frequently occurring alleles. That's frequency greater than 1% in the global population. And the second option will give the option of choosing from any of the alleles available for which we have algorithm available. And the third option is select from list of representative alleles from HLA supertypes. So we have a set of supertypes defined. So this is a representative from supertypes. And the fourth option gives automatically selects all the most frequent alleles, the set of 27 most frequent alleles. So you click that, it's automatically selected. And the last option is upload allele file. If you have your own custom list of alleles, you can upload that here, just same as what we have right now. So right now, let me select 2 alleles. So you can do the multi-select here. And going to the next step. So in the next step, we can choose the number of peptides. So select peptides to be included in the prediction. So the first one is apply default settings for low number of peptides. So that means here we are removing all the duplicate peptides. So if you have multiple sequences, same duplicate peptides are repeated, these duplicate peptides will be automatically removed, and only peptides of length 9 will be included here. And this shows how many 9mers are included in your sequences. And if you want a little bit more peptides, you just select the moderate number of peptides, which gives automatically, like it selects 8mers, 9mers, 10mers, and 11mers. And if you want more, it selects all the lengths from 8 to 14, and duplicate peptides will not be removed. And at the end, you have custom selection option of choosing custom selection, like you can select what all lengths you want, and if you can choose whether you want to keep or remove the duplicate peptides. And at the end, there's another option of conservancy analysis. So you have multiple sequences, and you want to have the results, the peptides in results that are conserved in only specified percentage of sequences. Then you can check this one here, and you get this option. So if you want to have peptides that are conserved only in 50% of sequences, you can choose this and select how many sequences you want. So I choose to have the conservancy analysis, and I want to use only peptides that are conserved in at least 50% of sequences.
A question, when it's showing the equals, is that in real time telling you how many 8mers, 9mers,
Yes, exactly.
10mers?
Yes,
Okay.
from your sequence.
Okay.
So the next step, 5th step, is where you select the method. So as of now, like now, we have all the methods available here. By default, we have the IEDB recommended. And for selecting the predicted peptides, we have different options as well. So for example, by default, what we recommend is to use the predicted percentile rank and the threshold of 1. So this will automatically select all the peptides that have percentile rank less than or equal to 1. So if you want to adjust that, like make it 5, you can do that. And the second option is select peptides based on predicted IC50. So you don't want to use the percentile rank, you want to follow the IC50, you can do that here. And by default, the recommended value is 500 nanomolar. And if you want to make it 1000, you're free to do that. And the next one is peptides based on MHC specific predicted binding. So we published a paper in 2013 in Journal of Immunology. So we found that if you use allele specific predicted binding affinity, that may be slightly better, than going with the predicted percentile rank. So we have these predicted affinity, the allele specific predicted affinity values only for this set of alleles. So if you are using this, predicting for this set of alleles, so we recommend using this one. Select peptides based on MHC specific binding threshold. So for more details are given in the help page. So for each specific editor showed that in one slide. So for each allele, there's MHC specific, allele specific binding affinity threshold.
Sorry, another question. Is that, 2 questions. Is that the list of the 26-7?
No, this is not that.
This
is where we can employ this method.
Okay.
So for a set of different alleles, we have got this threshold, MHC specific binding affinity threshold. We calculated that.
Okay.
So for this set of alleles, you can use this method. So, but if your allele is not here, you cannot use this method.
Yeah. And can this tool handle doing simultaneous MHC1 parameters and MHC Class II parameters? So where you're using the IC50 threshold of 500 for...
So that's for class 2, it's a different path. I will show you that.
Okay.
So when you use this recommended, allele-specific binding affinity threshold, this is for, corresponding to 75% of the immune response. So more details is given in this paper, 2013 paper. And the last and other options are like, if you want to get 2%, top 2% of the peptides, so you have like 250 peptides and I want to get the top 2% of peptide, you can do that here. So for example, I want to get 5% of peptides, so it will automatically calculate here how many peptides you'll get in the result. And similarly, you can specify the number of peptides. So I don't want to estimate the percentage, I just want top 50 peptides. You can specify that here, like that. So for now, let me choose the first one and let me select peptides, cutoff value 5. And now we go to the last step. Here you get all your input parameters. You can review that. Number of sequences 3, species human, class 1. These are the 2 alleles. And peptide length selected 9 mers, 10 mers. And peptide overlap. This is not applicable to class 1. It's only applicable for class 2. And conservancy analysis. You want to do the conservancy analysis and you have set the threshold of 50% of sequence. And the prediction method you're going to use is IEDB recommended, and peptide selection is based on predicted percentile rank, and cutoff is 5. And here there are 2 optional things. You can give a job name. So it's better to give a job name, because if you have some issues and you want to contact us, and if you can include that job name, it will be easy for us to track down, okay, what happened wrong. And then there's an option for providing the email. So if you provide your email ID, once the job is done, sometimes if you have more sequences, it may take long, so you don't have to keep waiting. So if you provide the email ID, there will be an email sent to your inbox that the task is done, and it will have the top peptides that's predicted. Once you have everything ready, you submit. So this is the results. So at the top, you can see these are the concise results, or these are the top peptides according to that we selected based on your input parameters. So you have 2, 3, 4, 5, 6 peptides that are below consensus percentile 5, and that are conserved in more than 50% of sequences. And you can download this table here, clicking this link, and in the next section it shows the complete results. So that's what you get right now. And in the next link, this link is for conservancy of peptides. So let me download. Opening. Okay, I'll show you that. Yeah. So this is the complete result. This is what you get right now. So using the tool that we have right now, this is the complete result. So in case you want to check the complete prediction results, you want the results binding prediction for all the peptides, you can download that using this link here. And the conservancy, and this is the conservancy. So this shows for each peptide in how many sequences this is conserved 100%. So this shows the sequence number and the peptide start and end positions, and whether it's conserved. Conserved means it's 100% identical. So as of now, we don't have the option to blast or finding similar peptides, but 100%. So this peptide is conserved in 2 of the 3 sequences that I provided. So it's 67%. You have the option to download that. And the third section is citation information. So you need to cite these papers in case you are using these results. And the fourth section is input sequences. So these are the 3 sequences that I provided, and other input parameters if you want to cross-check. So right now you saw the class 1, and let's see how you can do the same for the class 2. So here in the second step, I'm choosing class 2, human class 2. So the options are slightly different for class 2. So at first you have like this. In the third step you can choose the alleles. And the first one is like predict for custom allele set. So you can choose your alleles from the by default sets. So these are the most frequent alleles. Select from list of alleles, or you can do upload allele file, your own allele file, same as what we have right now. And so you can choose for DR, or you can do multi-select, or DP, like this. And if you want to choose alpha and beta chain separately for DP, you can do that here, like this. And if you want to reset alleles, you can click here. So right now let me just choose a couple of alleles from here. Okay. So, and the second option available are predict for pre-selected panel of alleles. So for example, here's the most frequent 26 alleles. If you want to do the promiscuous binder prediction using the most frequent 26 allele, you can use this one. And you can either select all the DR, DP, DQ, or if you want only one set, then you can do that here. And the third option is predict using pre-selected allele sets and methods. So this is where you can use choose the 7 allele method that I explained some time back. So you click this, automatically it will do everything. So you don't have to do anything manually, and use 7 set of 26 most frequent alleles. So you use this for doing the promiscuous binding prediction using the 26 alleles. So let me choose this one here. So similar to the class 1, there are different options for inclusion of peptides. So by default, apply default settings for moderate number of peptides. So what changes here is the desired number of overlapping residues. So by default, it's overlapping by 10 amino acid residues. So if you set it as low, it will change as number of overlapping residues at 8. So this number will reduce. Like previously it was like 12, it changed to 8. And then if you want high number of peptides, this is increased. This is kept at 10, but it will include the duplicate peptides. And you can choose the conservancy here. And similar to class 1, you have the option to choose your method, and different options available. It's also same as class 1 based on predicted percentile rank. Select peptides based on IC50, and you can adjust the threshold here. By default it's 10, that's what we recommend, and IC50 is 1000 for class 2. And here one option is select peptides based on number of alleles binding. So this is something that we don't have in class 1. So if you are doing the promiscuous binding prediction, so this is the option that you can use. So you have a set of 10 alleles, or you choose the 26 alleles reference set. Then you can select the peptides based on predicted number of binding alleles, and you can adjust the threshold here. And these two options are same. You can choose the percentage of peptides that you want, or number of peptides that you want. So let me keep it here. Let me make it 15. This is the result. So it's also same as class 1, and one difference here is that the only difference here is that you get an additional table here, non-redundant results or prediction results with redundant peptides with each sequence removed. So that's for this tool, the new TepiTool, we are using the post-processing. So I mentioned for removing the redundant peptides, we can use pre-processing or post-processing. So here we are using post-processing, so that just to make sure that we always capture the best predicted peptide. So that table actually provides all the peptides after removing the redundant peptides. And
So question for you about this TepiTool. So am I understanding correctly that if you wanted to do the promiscuous binder identification, you don't have to do it manually? It will output that directly?
yes.
Oh, that's awesome.
Yeah. This is the email that you'll be getting. So once the job is done, you will get an email and it will have the concise table. And it will also include, it's not implemented right now, but it should include the links for downloading the complete table. Yeah, and yeah, this is what I wanted to show you. So this is the non-redundant result. So that when you click that link, you get the post-processed result. So this is how it works. So I showed this slide before, but it picks the top peptide and eliminates all the redundant peptides from the same regions. So that's it. And any question? So we went through comparison of class 1 and class 2 tools. So there are some differences between class 1 and class 2 tool, and we went through that and we saw how the tool works, and we went through all the different recommendations and different versions of the tool. We have got online or web interface, and we have got standalone version and tools or API. And we saw prediction of promiscuous binders, and I explained the new 7 allele method that we developed, we identified recently. And we also went through the TepiTool. So we will be releasing this by December. We hope to, because the manuscript is under review. So once it's published, it will be available at tools.iedb.org TepiTool.